Yeah, thanks so much for the introduction. And yeah, it's really great to be here and to be here in person to give this talk. And the talks so far have been really amazing. So as you mentioned in the introduction, I have a lab now at the GMI Institute of Singapore. I've had my group there now for about a year and a half. And I'm excited today to present some of the new work that we've been doing, some unpublished work on developing new methods for analyzing single-cell ataxic data. So my lab is very, very interested in how DNA regulatory elements function. And I'm sure many of you are aware of the importance of regulatory elements. So just to emphasize this further, we now know through many years of GWAS studies that actually more than 90% of the variants that are associated with a polygenic trait are falling in these non-coding regions. And so really these variants are somehow changing the function of DNA regulatory elements, and that is linking to our risk for different polygenic diseases. And so understanding how these regulatory elements function is really critical to understanding our disease risk. And what we also know is that the function of these regulatory elements is really determined or correlates with very complex patterns of biochemical marks that sort of decorate the DNA. So we know, of course, that the DNA is not just a naked piece of DNA in the cell. It's very highly packaged into a three-dimensional structure and wrapped around nucleosomes that have different post-translational modifications. And these marks can kind of give us a window into the activity of that regulatory element. And so this includes things like histone modifications and the DNA accessibility. And so if we want to understand how these regulatory elements function, then it's really critical that we can actually measure these biochemical states of the chromatin in the cell. And so, we've been very fortunate over the past several years now that actually there are many different experimental assays that have now been developed that enables us to measure different aspects of these chromatin states now with single cell resolution. And some of them are here. This is not an exhaustive list, but just to illustrate, there are methods for measuring chromatin accessibility through single cell ataxic methods, methods like single cell cut and tag for looking at histone modifications in single cells, different methods for looking at DNA methylation or three-dimensional chromatin structure. And more recently as well, there are other methods that have been developed that actually enable you to measure not just one aspect of chromatin state, but potentially several. And one of these is the method called SC-NTT-seq that we developed a couple of years ago now. And this enables you to measure multiple different histone modifications from the same cell. So we actually have a fairly extensive experimental toolkit now that enables us to measure what the chromatin state is in individual cells. But really measuring these states is just the start, right? Actually, the computational analysis of these data is still a major challenge. And we've done a lot of work on this aspect as well. One of the software that we developed is this package called Signac, which is an R toolkit that's designed for single cell ataxic analysis. And what this enables you to do is take the raw data, which in this case is essentially what's called a fragment file. This is essentially the list of all the sequenced pieces of DNA from your experiment, as well as where they map to in the genome and what cell they came from. And so from this file, you can run all the different downstream analysis tasks that you need to do for ataxic analysis, which includes things like peak calling or quantifying your peak by cell count matrix and computing different QC metrics and doing dimension reduction and all of these different things. So the raw data is really this fragment file. And then in our software, you can use that as input to do all these other tasks. And so, this software enables you to do a lot of things and is fairly broadly used now, which is really amazing. The problem that we're starting to have now is that actually, because the development of this software started now more than 5 years ago, the data sets have actually grown pretty substantially in size now. And the data is approaching the limit of what the current software can actually handle. So it's causing some headaches now in actually scaling the analysis of single-cell ataxic data to sort of keep up with the improvements in the experimental side. And so this is, there are really two challenges that I want to focus on today. The two major challenges in single-cell ataxic analysis. The first is this problem of scalability, which I just sort of mentioned. And we sort of run into this problem when analyzing single-cell ataxic data much faster than you would run into if you were looking at single-cell RNA-seq, because the number of features that we measure in single-cell ataxic is much, much larger for transcriptional assay. So for your single-cell ataxic experiment, you might have 200,000 peaks. And for your RNA-seq, you may have 20,000 genes, or you may subset that to 2,000 variable genes, right? So the matrix is much bigger, which means you hit these scalability issues faster. And then the second challenge is that still we really don't have a set of reusable features for ataxic analysis. So if you do a single-cell RNA-seq, you know what your genes are, you always quantify the same genes, right? But we don't really have the equivalent for ataxic, and so the current workflow for analyzing this type of data really starts with doing de novo peak calling on each data set. And that's kind of fine for a single project, but actually becomes a major headache when you start to try to look at published data and sort of aggregate data from many studies and reuse data. They become very difficult to compare. And just to illustrate this point a little more, here I downloaded 5 different single-cell ataxic datasets for PBMCs from the 10x Genomics website. And I'm showing the peaks for these 5 different datasets here. So they're shown as these sort of bars for a randomly selected region of the genome. So these are 5 different datasets, all from the same tissue, profiled by the same company, processed using the same software. But you can see that in some of these datasets, some peaks are just completely missing, right? They're not detected across all the different datasets. And so say we wanted to download these 5 datasets and compare the DNA accessibility in one of these regions, we start to run into problems because each of these bars here, these peaks, corresponds to a row in the count matrix. And so some of these datasets simply don't have those rows, right? So you're comparing something, some measurement in some datasets to no measurement in others, right? So it becomes difficult. But you might say, okay, many of the peaks overlap, right? They look like they're fairly consistently identified. And yeah, it seems like that in this zoomed out view, but if you zoom in, you also notice that the precise start and end of the peaks, even in these peaks that overlap fairly well, varies across the different data sets because these are produced by independent peak calling on every data set. So then you're still introducing this sort of variability across data sets purely based on where did the precise start and end of the peak happen to be. Which is not how you would really ideally want to run this sort of comparative analysis. So while this seems like a somewhat trivial problem, it actually can be a major barrier to using public data, reusing data, because actually in many cases, the raw data that you need to re-quantify these count matrices is not available. So or either it's not available or it's behind some restricted access database. And so then there may be a fairly substantial barrier to getting access to that data. And this is something that in my lab we've sort of come up against many, many times, is trying to get data sets from different studies of Geo, for example, and the peaks are different and the raw data may not be there. So it becomes difficult, especially if you're trying to aggregate a lot of information together for example, training a model. So we set out to try to see if we can address this problem and create some universal set of features that we can use for single-cell ataxic analysis. But we started looking into this and looking at the literature, and of course, there's been quite a lot of work done on this sort of problem already. And the first sort of group that has looked at this is from ENCODE. So they defined this set of just over 1 million what they call candidate cis-regulatory elements, these C-series. And this is from many thousands of different epigenomic experiments that have been run on human samples, and they also have for mouse, but here I'm focusing on human. So they come up with this list of candidate cis-regulatory elements. Essentially, the DNase hypersensitivity sites from different experiments. And then more recently, and this paper is still on bioRxiv, but from Xuegong Zhang's lab, they developed what they call a consensus set of attack-seq peaks, really motivated by the same problem. But here they looked at over 600 different bulk attack-seq experiments from different human tissues, and they come up with this set of around 1.2 million consensus peaks. And so, we didn't want to reinvent the wheel, obviously. We can just combine these, and so we combine these different sets, and we get a set of around 1.5 million regions, because of course many of these different regulatory elements overlap. So we have this set of 1.5 million regulatory elements, right? And so you might say, okay, then that universal feature set problem is not that hard to solve. You just merge some bed files, right? And maybe, but in solving that, you really make the scalability problem much, much worse, because as you add more and more features that you're quantifying, the scalability gets much more challenging. So if we look at the situation that we have for RNA-seq analysis, right? In this sort of blue rectangle, this could be our count matrix. So we would have, let's say, 20,000 genes by some number of cells. Then we can do feature selection and subset that to a set of variable genes and do dimension reduction. For our typical single-cell attack-seq analysis, we have 1 to 200,000 peaks is typical. So we have a much larger matrix, and then we have to do dimension reduction on that. So it's already more challenging, right? Much bigger matrix. If we then move to using this universal feature set, now we have a giant matrix of over 1.5 million regions, and it's running off the page, right? It's just too big to work with. So while you've sort of solved one problem, you make the other problem of scalability much worse. So while you can kind of define these features and they're universal, good luck kind of reading this into your R session and doing anything with it, right? So the observation that we had, which is I'm sure kind of obvious to a lot of people also, many of the regulatory elements are very highly correlated with each other. We don't have 1.5 million totally independent regulatory elements. A lot of these are co-regulated by shared factors, and they'll have high co-accessibility in cells. And so we thought, okay, maybe we can leverage this redundancy and try to sort of collapse this redundancy in the data set and reduce the number of features that we're looking at in that way. So we aim to find these groups of co-regulated DNA elements and sort of collapse these into different modules, and then we could run an analysis based on the accessibility of modules rather than individual regulatory elements. So this is what we did. We aimed to find these regulatory element modules, and we started out by leveraging the massive amount of epigenomic data that is out there, mostly from ENCODE. So what we did is we quantified for each of these 1.5 million regions the signal in a set of over 4,000 different experiments from ENCODE on human cells. And then we can do a sort of dimension reduction and clustering based on that. And so here, each point is a regulatory element. But this really gives us a fairly broad level separation of different types of elements. So it separates things like promoters from enhancers and CTCF sites. But we wanted still, much more granularity than that, right? So we sort of use that as a starting point. But then within each of these broad level clusterings of the regulatory elements, what we do is we compute a score based on two different things between each pair of cis regulatory elements. So firstly, what we did is we assembled this collection of cell type accessibility profiles from a lot of different published single cell ataxic studies. So we have profiles for over 500 different cell types. And so using this collection of data between each pair of cis-regulatory elements that are within the same broad level clustering, we compute the co-accessibility across these different cell types. So that gives us a co-accessibility score. And then we looked at a set of 45 very high quality, deeply sequenced Hi-C data sets that were also generated through ENCODE. And we used these data sets to compute a physical contact. And then we just combine these, we compute a co-accessibility by contact score for every pair of these regulatory elements. So once we have those scores, we create a graph, a weighted graph. So here each node is a cis-regulatory element, and the edge between nodes is this co-accessibility by contact score. So if they have a high edge weight, that means that those two cis-regulatory elements have high co-accessibility, and they also have physical contact measured by these Hi-C experiments. So we have this weighted graph. Then all we need to do is do some graph-based clustering or community detection algorithms, and then we have these clusters of highly connected nodes, which are basically highly similar cis-regulatory elements. And so we have these regulatory element modules, and we named these REMO for the regulatory element modules. So using this approach, we sort of collapse our total number roughly in half. So, the first thing that we wanted to look at, of course, is do these groupings of regulatory elements seem to make sense? And so what I'm showing here is just a few different cell types from the immune system and their accessibility. And then along the bottom, we have these cis-regulatory elements, and they are colored by the module that they belong to. So here we see two elements that are close together. They're both accessible in the NK cells, and they're in the same module. And then some others that are accessible in different cell types and are not in the same module. In this example, these two are clearly co-accessible in the monocytes and are put into the same module and are separate to these other regulatory elements in between. Then we have other examples where the regulatory elements are much further away in the genome. So here we have some elements that seem to be specific to the T cells, particularly naive T cells. And in this case, they're around 60 kilobases away, and they're still grouped together in the same module. And then we can look more sort of systematically genome-wide. If we take a single cell data set and we compute the co-accessibility among the single cell data for modules, cis-regulatory elements that are within the same module versus in different modules, we can compare their level of co-accessibility using Pearson correlation. And we can see that in sort of orange color, this is the distribution of Pearson correlation for elements that are within the same module, and it's much, much higher than those that are in different modules, right? So that makes sense. Now we have these regulatory element modules, and we want to use these as new features that we can quantify in our single cell ataxic data. But the next problem that we sort of run into is that the current software that we have from this SYNAC package that I mentioned at the beginning, the software that we have to quantify our features in the single cell ataxic data really doesn't scale well with increasing number of features. So here, if we look at a data set, this is just a 9,000 cell data set, and we can look at the runtime and memory usage for increasing number of peaks that we want to quantify from 0 to 1 million. You can see that the memory and runtime kind of goes up linearly, right? And for this small data set, it's not the end of the world because, maybe it takes 15 minutes to run. But if we want to scale this to many millions of cells, then this actually becomes a problem, right? So what we did is we actually developed a new software toolkit that we called Fragment File Toolkit, FragTK, that's implemented in Rust, which is a different programming language, and enables running this matrix quantification, so building some region by cell matrix from your raw data. And this is also nice because it's very simple to install. You can just download a binary from GitHub and just run it. You don't have to do anything else. So this is on our GitHub now. And if you look at the scalability of this software compared to the R implementation in Synyac, you can see that it scales much better now with increasing number of features. The runtime really doesn't change much. The memory usage is much less. And importantly also, we implemented another quantification method that enables you to quantify groups of genomic regions as one row in your matrix. So this would enable you to quantify like a module of features as one row in your matrix. And of course, we can also look at how this scales with different number of cells. So if you just wanted to use this software as a sort of replacement for your building your count matrix for your single-cell ataxic analysis, here we looked at two different data sets, one for 9,000 cells and one for 650,000 cells. And if you look at, as you downsample the cells, how does the runtime change? You can see, the Rust software does much better. So just taking 400,000 cells as an example, it's around 4 times faster, 3 times less memory. So you can use this as just a sort of drop-in replacement for doing your count matrix quantification for single-cell ataxic analysis and save some time. Okay, so now we have these regulatory element modules, and we have some better software that enables us to quantify those in single-cell data. So what we wanted to do is, of course, apply this to some single-cell data and see how does it perform in comparison to, say, quantifying a peak by cell matrix. So the way that we did this is we took multi-ohm data, where in each cell we have measurement of gene expression and DNA accessibility. And what this enables us to do is we can annotate the cells using the gene expression information, and then we can process the DNA accessibility data in 2 different ways. So either using the REMO modules or using the more standard peak by cell matrix quantification. And then we can ask, which has better agreement with the RNA-based cell labels. So that's what we did. And here you can see UMaps created from either quantifying these REMO modules or peaks, and they look similar. They both do a fairly good job of separating out the different cell types in the data set. Here again, the cells are labeled by their RNA-derived cell type. But we can look a little more quantitatively about what has better agreement with the RNA-derived cell label. Is it the clustering from the REMO modules or the peaks? And if we look at that using adjusted rand index, actually the REMO modules has slightly better agreements. But really they both do fairly well. But a key observation that we had is actually the performance of the modules. If you look at doing this dimension reduction with different numbers of modules. So if we rank the modules based on their activity in the data set, and then we run the dimension reduction using different numbers of the top ranked modules, we can see that the performance really plateaus quickly at around 20,000 modules. So then, which makes sense actually, because these modules, this universal set of cis-regulatory elements covers really every possible tissue from the human body. So if we're just looking at the immune cells, a huge number of these are going to be not necessarily active, right? So we don't have to include those in the analysis. So now we can actually subset our features. Instead of 1.5 million, we have 20,000 modules now. And so that's really key for scalability. So these results, this UMAP and the adjusted rand index, is actually using just the top 20,000 modules. But the benefit in terms of scalability really only applies if we can do some disk-based subsetting of the features, right? Because we don't want to create this giant matrix and then have to load that into memory and subset it into the top 20,000. That just doesn't make sense. So, what I showed so far is we have our raw data as this fragment file, and then we have this fragtk software that we developed combined with our REMO modules that we identified. And using these, we can create our sparse matrix, a REMO module by cell matrix, which is a sparse matrix that lives on disk. as a MTX file, right? It's just a sparse matrix format. So what we did next is we created a new software program that we called Sparks, which is a disk-based sparse matrix statistics and subsetting program, right? So this can very efficiently compute statistics on the matrix and perform subsetting of that matrix on disk. And so using that, you can very efficiently subset this larger matrix to a smaller one. And then using that smaller matrix, we can then load that into our memory and do downstream analysis. So then we sort of envision this running as these steps running on disk, and then you load a very small subset of your data into memory and do downstream analysis. And just to give a ballpark figure, for a 25 gigabyte matrix file, this uses about 4 megabytes of RAM to subset that matrix. So now that we have this, sort of improved scalability, we wanted to benchmark this a little more as well. So we found a much larger single-cell ataxic data set from Jay Shenjurio's lab. This is 770,000 cells profiled using single-cell ataxic from different human fetal organs. And so what we did here is we took this 700,000 cells and downsampled to different numbers of cells, and then just benchmark how long does the dimension reduction take using the REMO modules or the peak matrix. And so, unsurprisingly, because the matrix is much smaller, you have a fairly substantial gain in the runtime and memory usage, about 3 times faster, 40% less memory using the REMO modules. And then, the other really important point actually is that because the matrix size for the REMO modules is much smaller, and there's actually a limit to how big a sparse matrix of a particular class, but the more commonly used class, can be in R. When you use the peak matrix, you hit that limit at just over 400,000 cells. And so once you surpass that limit, you actually can't load the matrix into your R session anymore. So then even if you had terabytes of RAM, you really can't do much because you simply can't construct that matrix in memory. So when you're using the REMO matrix instead, you can scale to the size of the whole data set quite comfortably without reaching that limit. Okay, and so I started out also talking about that we want this to be a universally applicable set of features for single-cell ataxic analysis. So earlier I showed some analysis on PBMC. That's kind of at the end here. But then what we've looked at also is collecting many different multi-ohm data sets from other tissues. This is mostly from stuff that's on ENCODE or from the 10x Genomics website. But here we have brain, heart, jejunum, liver, lung, muscle, pancreas. And these are all the same sort of analysis that I showed earlier, where the cells are annotated using the gene expression information. And then we can compare how much does the cell label agree when we process the cells using either a peak matrix or the REMO matrix. And so, you can compare the UMAPs here. It's a little hard to, compare how the UMAP looks. But if we look at the adjusted rand index, which is more quantitatively looking at this agreement, actually across all these different data sets, we see that the REMO quantification does do slightly better in terms of agreement with the gene expression-based clustering. But really, I think both REMO and peaks do quite well in separating out the different cell types. I think the advantage is that in each of these cases, for the peak matrix, you have to call a different set of peaks for every one, and it's in these cases, 1 to 200,000 peak regions, and so you have to change the features in every time. Whereas for REMO, we can just reuse the same set of modules across all these different tissues and get consistently good results. And as I showed in the previous slide, the performance is much better in terms of the scalability. So one last thing that we looked into, which I think is quite interesting, is that you can actually use these modules to help interpret your data as well. Because in clustering these cis-regulatory elements, we downloaded a ton of epigenomic data. And then we started thinking, okay, we can actually annotate these modules with the types of experiments that have the highest signal for active histone modifications, for example, and ataxic experiments, different active marks. So, we annotated now each of these modules with a list of terms that we derived from the ENCODE experiments that had the highest signal in those cis-regulatory elements. So, for example, REMO 1 might be something like T cell, and 2, adipose tissue, and things like that. So we're trying to sort of get some metadata associated with these different modules. And then we can use that to help interpret the single cell data. So if we, for example, we can look at cluster 2 here. This is the PBMC dataset that I showed earlier. If we look at cluster 2, we can find all the different modules that are specific to that cluster. And then we can look at the list of terms that are annotated for those modules, and we can do an enrichment test, sort of an ontology enrichment analysis, and find these enriched terms. So in this case, the enriched terms are shown here. They're all T cell related, and particularly naive T cell related. These are actually all of the significantly enriched terms, so they're all related to T cell. And then because in this data set we have multi-ohm data, we can look at the gene expression, and we can see that LEF1, which is a marker of naive T cells, is expressed in cluster 2. So it kind of matches our enriched term analysis. We can look at other clusters. So here we do the same thing for cluster 8. The only enriched term that comes up is for natural killer cell. We can look at GNLY, which is the natural killer cell marker, and it's consistent with that. We can look at the cluster 7 here as well. All the enriched terms are related to B cells, and then this cluster also expresses B cell marker genes. So this is just another way that we can start to interpret the single cell ataxic data. And I think is particularly useful in cases where we don't have multi-ohm data. So then we wouldn't have the gene expression profiles to look at. But we have many cases where you have only single cell ataxic data, and then you can start to sort of use these annotations to help interpret the data. I think it's not the one and only thing I would do to annotate the cells, but can be sort of another tool in the toolkit when you're exploring your data. Okay, so with that, just want to summarize the main things that we've developed here. We've developed this new set of features that we call REMO for single cell ataxic analysis. And this is both universally applicable across different human tissues with consistently good performance, and is much more scalable compared to using the traditional peak matrix quantification. And then this is sort of accompanied by 2 new software tools that we've developed that are both implemented in Rust. FragTK, which is for quantifying your count matrix, and then Sparks, which is once you have that count matrix, you can efficiently compute statistics and subset that matrix on disk. And these are both available on our GitHub page. So with that, I'd like to thank all the members of my group, especially Krisania Lim, who has led the development of the REMO project. And yeah, so with that, I'd like to stop here and be happy to take questions. So thank you.